All right, everyone, welcome back. In this lab recording, we're going to begin covering the skeletal system. Now, we will be covering 3 topics that will deal with the skeletal system, and this will be the first of the 3. This presentation will serve as your introduction to the skeletal system. The other 2 topics will separately cover the axial skeleton and the appendicular skeleton, which will refer to the 2 different branches of the skeletal system in general. The axial skeleton in particular includes the skull, the vertebral column, and the thoracic cage. And the appendicular skeleton includes the 2 shoulder girdles, the 2 arms, the 2 legs, and the pelvic girdle. It will be in our second 2 topics when we will individually learn all of the bones of the skeletal system and also some of the bone markings that can be found on each of the bones. But before we start to master the components of the skeletal system, we first must discuss some of the theory behind the skeletal system, the classification of the skeletal system, and the different general bone markings that we will find on our bones. So we're going to go ahead and get started by first explaining the term articulation. I will be using this term quite frequently throughout the course of this presentation, as almost all of our bones will be articulating with other bones. An articulation refers to a joint, where 2 bones come together. Any area where 2 bones contact each other and form a joint is an articulation. We can see articulations up here at the shoulder, some articulations here at the elbow, and many articulations that can be found in the wrist. Every single joint will be composed of 2 bones articulating. We will learn which bones articulate with what other bones in the coming lab recordings. So the first thing that we need to discuss in regards to bone is what bone truly is. Bone is a type of connective tissue, specifically one of our types of hard or supportive connective tissue. Remember, connective tissue is composed of cells and a non-living extracellular matrix. And the extracellular matrix of bone is a substance known as osteoid, which is what makes bone very, very hard and rigid. In general, there are 2 types of bones, specifically compact bone and spongy bone. Just as it sounds, compact bone is very, very dense, and it's typically found on the outside of our bones. It will often serve as a protective shell that will wrap around the outside of our bones and protect the structures that can be found within. Histologically, compact bone will look a little bit different than spongy bone, and we will see what each of these types of bone look like under the microscope as we progress through the skeletal system. Spongy bone, on the other hand, is what we see found in here. Spongy bone is given its name because it looks like a sponge. But make no mistake, spongy bone is not squishy like a sponge is. Spongy bone is very hard and rigid, and it will serve different purposes than compact bone. Compact bone was very, very dense, and it provides that protective shell for the outside of our bone. Spongy bone, on the other hand, serves to lighten bone and also provide structural support to our bones as a whole. I will be further discussing compact bone versus spongy bone throughout the remainder of this lab recording, but I want us to have a little idea of how these 2 types of bones differ from each other. The general organizational differences that we will see between compact and spongy bone is that compact bone is often found on the outside of bones, while spongy bone can be found on the inside of bones. As we move into the classification of our bones, we will come to see this to be true. So one thing that I tend to focus on specifically with anatomy and physiology as a whole is the classification of our different systems. There's so much information that we have to learn in anatomy and physiology, and it can be quite overwhelming if we don't have any way to organize all of this information. So the classification of different systems serves to keep us organized and to help us understand the differences between these structures. So bones are no different. We would classify bones based on their shape, and there are 4 groups of bone classifications that we will see. We have long bones, short bones, flat bones, and irregular bones. And each of these classifications is fairly logically named. Long bones will in fact be much longer than they are wide, but they will have some other characteristics that are extremely important to note. Short bones will be rather cube-like in their shape, and all of their sides will be roughly the same length. Flat bones, as they sound, will generally have a wide and slim structure. Based on their flat shape, they do typically tend to be curved as well. And lastly are the irregular bones, which include all of the bones that do not fit into one of these other groups. Some irregular bones are going to have short parts, but also may have flat parts. So we group these in this fourth category of irregular bones so that they can have a home as well. So the first classification of bones that we're going to break down are long bones. Long bones have a very unique and specific structure, and you may have learned the long bone structure in your lecture class as well. As their name suggests, long bones are in fact much longer than they are wide. However, this is not the determining factor for why bones get classified as a long bone. Long bones in particular can often be found in the arms and the legs. And on top of being longer than they are wide, they will have two very enlarged ends and a central skinny shaft. These enlarged ends that are separated by this central shaft are in fact the requirements for a bone to be classified in this group. We are next going to go into the structure of the long bone, but I want to also state that long bones will have compact bone on their outsides and spongy bones on their insides. We will see this with the structure of the long bone. So I probably should have said this earlier, but all bones are considered organs. We must think back to the levels of structural organization to define what an organ is. Organs are made up of more than one type of tissue. And bones are a type of connective tissue, but they'll have nervous tissue that can be found in them as well. And if we consider the extreme vascularity of our bones also, they'll have epithelial tissue found in them as well. So all bones in fact are considered organs based on the presence of many different types of tissues found within them. This is not just unique to long bones, but what is unique to long bones is this structure. Every long bone is going to roughly have the same components that make it up. And it is this structure that we use rigidly to classify bones as long bones. In particular, there are two main regions that can be found on long bones. We'll find two epiphyses and one diaphysis. Think back to how I mentioned those two enlarged ends and the central shaft. The epiphyses refer to the enlarged ends, and the diaphysis refers to the central shaft. We will have other substructures that can be found within the epiphyses and the diaphysis, such as a medullary cavity, articular cartilage, and the epiphyseal line, which will separate the epiphyses from the diaphysis. We'll also have several connective tissue membranes, namely the periosteum and the endosteum, that we're going to see present on different parts of our long bones. Let's move to the next slide, where we will see the structure of the long bone and all of its parts that you'll need to be aware of. So first and foremost, you'll see that the long bone structure has these two enlarged ends and this central skinny shaft. We'll go ahead and start with the diaphysis in particular, and that is the central skinny shaft. You can see this middle portion is labeled the diaphysis, and on either end we'll find the epiphyses. We'll have both a proximal epiphysis and a distal epiphysis, based on the proximal and distal sides of the bones. Both the epiphysis and the diaphysis are separated by this structure known as the epiphyseal line. Oftentimes, the epiphyseal line is referred to as our growth plate, and it is at the epiphyseal line that bone elongation will take place as we age. Unique structures will be found associated with the epiphyses and the diaphysis. Let's go over the structures associated with the diaphysis first. In particular, the diaphysis is going to contain a space. There will not be spongy bone found in the center of the diaphysis. This space is known as the medullary cavity, and inside of this cavity we're going to contain a very important structure. This yellowish substance that's found inside of the medullary cavity of the diaphysis of our long bones is known as yellow bone marrow. Yellow bone marrow is in fact adipose tissue. So inside of the medullary cavity, we will find stores of adipose tissue. We'll find that the walls of the diaphysis are made up of a very, very strong and thick layer of compact bone. As we come up here to the epiphysis, this will also be lined with compact bone, but it will not be as thick as it is found down here at the diaphysis. The other structure that will be found inside of the medullary cavity that's lining the medullary cavity is known as the endosteum. The endosteum can be described down here. This layer of connective tissue will line the inside of the medullary cavity and contain the yellow bone marrow. The endosteum is not the only connective tissue layer that we will see on the diaphysis. In fact, we're going to have another structure that wraps around the outside of the diaphysis known as the periosteum. Peri means around, and osteo is indicating that we're talking about bone. So the periosteum is the connective tissue layer that wraps around the diaphysis of our long bones. So from superficial to deep, we have the periosteum that wraps around the diaphysis and a very thick layer of compact bone. We'll find the endosteum lining the inside of the medullary cavity and the yellow bone marrow that's contained within that connective tissue layer lining the inside of the diaphysis. This is the structure of the diaphysis in particular. But as we move to the epiphyses, we'll see that there's a different structure that can be found at each of these ends. As we know, the epiphyseal line will separate the diaphysis from the epiphysis. But the epiphysis does not have a medullary cavity. In fact, we'll find spongy bone in the central portion of the epiphysis. We've zoomed back into spongy bone to again show you what this looks like. These irregular spicules or bridges of bone will grow in random directions and connect with each other to provide a very structurally sound network. In particular, these spicules are referred to as trabeculae. So we would more accurately say that spongy bone trabeculae can be found inside of the epiphyses. One other structure that is not labeled on this slide that I do want you to be aware of is the red bone marrow. Red bone marrow will be found inside of the spaces between the spongy bone trabeculae, and that will be stored up here. I've moved to this slide just to show you the contrast between red bone marrow and yellow bone marrow. We have switched bones here. We're now looking at a different long bone, but the concepts that we've discussed on the last slide still do apply to this bone. This bone will have two epiphyseal lines, as you can see, but it will still have its medullary cavity, and we'll still see the epiphysis up here. You can see the reddish color is indicating the red bone marrow that is found between the spongy bone trabeculae inside of the epiphyses. And the yellow bone marrow can be found down here in the medullary cavity. The last component that we will see in the structure of the long bone is this articular cartilage that wraps around the outside of the epiphysis. Now, in general, you can see that the periosteum of the diaphysis will make its way up here to the epiphysis in some places. But for the purposes of my quizzes, you can state that the articular cartilage is found on the outside of the epiphysis, and the periosteum is found on the outside of the diaphysis. While there is a little bit of gray area in here, so that we're clear on the quizzes, you can make this distinction between the articular cartilage of the epiphysis and the periosteum of the diaphysis. This articular cartilage is named because this will cover the portions of the bones that will articulate with other bones. Long bones in particular will articulate at their epiphyses, and that's where this articular cartilage gets its name. So make sure that you have a very good understanding of this long bone structure. While long bones are named because they are longer than they are wide, it is truly this long bone structure that distinguishes long bones from other types of bones. We will have other bones that are in fact longer than they are wide, but they will not have an epiphysis and a diaphysis, so we cannot classify them as long bones. Our next classification of bones are the short bones. Short bones, as I mentioned, are cube-like in their shape. Their length, width, and height will roughly be the same. We will typically find short bones in the wrist and in the ankles. We'll have 8 carpal bones and 7 tarsal bones, which are all examples of short bones. And keeping with the trend of our skeletal system, short bones will have spongy bone in the center, and they'll be surrounded with a compact bone shell for protection. We could say that there is in fact a 5th classification of bones known as sesamoid bones. But sesamoid bones in this presentation are grouped under our short bones because they will closely resemble short bones in their shape. Sesamoid bones can be described as any type of bone that is enveloped in a tendon. There are several sesamoid bones that can be found in the body, but the most prominent sesamoid bone that we're going to see is the patella, which is also known as the kneecap. You can see the patella shown right here. This bone is classified as a sesamoid bone because it's wrapped up in a tendon known as the patellar tendon. This tendon will connect to muscles on the lower side of the leg, but also connect to muscles on the upper side of the leg also. Flat bones, as they sound, are flat in their shape, but they oftentimes do tend to be slightly curved. The bones of the cranium, the sternum, and ribs are all examples of flat bones. Technically, the sternum is a little bit longer than it is wide, but its flat shape and the lack of an epiphysis and a diaphysis denote this bone as a flat bone. The ribs are another flat bone that are often confused with long bones. The ribs are in fact longer than they are wide, but they don't have an epiphysis and they don't have a diaphysis. So we will classify ribs as flat bones. Just as we saw with short bones and with long bones, flat bones will have a compact bone shell with spongy bone in the center. Our last bone classification refers to our irregular bones. These are all of the bones that don't fit into any of our other groups. 2 examples of irregular bones are the vertebrae and the coxal bones, which are also known as the hip bones. The vertebrae in particular have a couple different parts to them. We have this cube-like portion, which would likely have been classified as a short bone if it was by itself. But we have this other flat portion that's sticking off the back. And so therefore, vertebrae can't really be classified as flat bones or short bones. So we stick these in our miscellaneous group known as irregular bones. Irregular bones will also have spongy bone trabeculae in their center and compact bone on the outsides. So now that we've discussed the different classifications of bones, it's time for us to go into the different types of bone markings that we will see on our bones. Bone markings can be separated into the three groups that we can see labeled on this slide. We have bone markings that are projections for muscle, tendon, and ligament attachment, bone markings that will serve as projections to form joints, and we'll have bone markings that will serve as depressions and openings for different vessels to course through. As we have not learned the individual bones of the skeletal system yet, our understanding of bone markings will likely remain incomplete until we learn the axial and appendicular skeletons. But what I want you guys to specifically be focusing on as we go through the bone markings is defining what these bone markings refer to and classifying them into their respective groups. There are lots and lots of bone markings that can be found on the skeletal system, and some structures will have very similar names. But based on the type of bone marking that we're looking at, we can classify it based on its role. Is it projecting from a bone to help in joint formation? Or is it perhaps projecting from a bone to give a surface area for muscle, tendon, or ligament attachment? These are the concepts that we want to be primarily focusing on as we go through the bone markings, and then when we start going over the axial and appendicular skeletons, you'll start to solidify your knowledge of specific bone markings and where they can be found. So we will first cover the group of bone markings that serve as projections for muscle, tendon, and ligament attachments. On this slide, you can see 4 words that will describe 4 different types of bone markings. Specifically, we have tuberosities, crests, trochanters, and lines. All of these structures are bone markings that will serve as an attachment location for muscle tendons or ligaments. Each of these structures is named based on their shape. And after we define each of these, we'll see some examples that can be found on the skeleton. So first and foremost is a tuberosity. This is one bone marking that we're going to see very, very frequently. A tuberosity is a rounded prominence that tends to be a bit larger. Tuberosities do differ in their sizes. However, compared to some of these other bone markings, as a group, tuberosities refer to processes that stick out of our bones. 2 good examples of tuberosities can be found on the radius and the ulna. These are the 2 bones that can be found making up the forearm. The radius bone in particular is found here, and it contains a tuberosity known as the radial tuberosity. This is a large, rounded prominence that sticks out of this bone, so we give this structure the name the radial tuberosity. We'll also see that this bone is named the ulna, and the ulna will have its own tuberosity known as the ulnar tuberosity. So with these 2 structures in particular, they're named based on the bone on which they can be found. While the radial tuberosity does look like it's a little bit bigger than the ulnar tuberosity, the size is not necessarily important. What is important is that both of these are processes or rounded prominences that come out of our bones, and they will both serve as attachment locations for muscle tendons or ligaments. Crests are the next bone marking that we will see. These are prominent ridges that can be found on bones. One great example of a crest that can be found on the skeletal system is the iliac crest that will be found on this portion of the coxal bone known as the ilium. We give it the name the iliac crest because of the bone that it's found upon. One other bone in which we will see a crest is here on the femur. The femur will stretch from the hip all the way down to the knee. And here we will find what's known as the intertrochanteric crest. This again is a narrow, prominent ridge that can be found on a bone. It's much shorter than the iliac crest, and it's not as curved as the iliac crest, but it's still a narrow ridge that fits into the group that we're discussing. Trochanters are very, very large and unique processes that can specifically only be found on the femur. In the human body, we will specifically find the greater trochanter and the lesser trochanter. Technically, we have 4 trochanters in the body because we have a right femur and a left femur. But the greater trochanter and the lesser trochanter are the only 2 trochanters that we have in the body, and all trochanters can be found on the femur. Trochanters do tend to be a little bit larger than tuberosities. But aside from this distinction, tuberosities and trochanters both fit within the same group. Our last bone marking for this slide refers to lines. Lines are very similar to crests in that they are narrow ridges, but they tend to be less prominent than crests are. So we're naming these two structures based on how prominent the ridge is. One great example of a line is the linea aspera that's found on the backside of the femur. We'll also have these supracondylar lines that can be found on the backside of the femur that are formed from the linea aspera also. We can see at the proximal side of the femur we'll have the intertrochanteric line and the intertrochanteric crest on the back side. The crest tends to be a little bit more pronounced, so that's why it gets this name rather than this name. Once again, I will say that I don't necessarily want you to be able to distinguish crests from lines, but I do want you to be able to distinguish all of these structures from the projections that will be helpful for joint formation. I cannot stress enough the importance of these three different groups in classifying each of these bone markings into their respective groups. Tuberosities, lines, trochanters, and crests are not the only structures that will serve as projections for muscle and ligament attachments. Tubercles, epicondyles, and spines are three other projections that are used for muscle, tendon, and ligament attachment as well. So these will be found in the exact same group. Let's start with tubercles. Tubercles are very similar to tuberosities, but these tend to be a little bit smaller than tuberosities are in general. That is not always true, as there are certain bones that have very large tubercles. So the discrepancy between tuberosities, tubercles, and trochanters is not necessarily important, but I do want you to understand that this structure is very similar to tuberosities and trochanters in particular. One great example of a tubercle is the adductor tubercle that can be found on the distal side of the femur. I know that we haven't spent much time identifying different bones, but this is in fact the distal side of the femur, and the adductor tubercle can be found right here. This is a very small, rounded prominence that can be found on the bone, and certain muscles will come in and attach to this location. Epicondyles and spines are our last two projections for muscle, tendon, and ligament attachment. Spines are pointed and very skinny projections that can be found on our bones, and epicondyles are raised areas near condyles. Condyles are in fact one of our projections for joint formation, and we'll learn about this one very, very shortly. But before we get into this group, let's go and look at a couple examples of epicondyles and spines. Using the same picture as we did with our tubercle, on either side of the distal end of the femur, we'll find the medial epicondyle and the lateral epicondyle. Epicondyles are by definition raised areas near condyles, and so we'll actually see both a medial condyle and a lateral condyle over here, which we'll discuss in the next group. But epicondyles are these larger raised areas near the condyles, and we can see those highlighted in green. This slide shows us the humerus bone, which stretches from the shoulder down to the elbow. On this bone, we'll see that we have a greater tubercle and a lesser tubercle that can be found here. Again, tubercles' definition are small, rounded prominences, but the greater tubercle is in fact bigger than some of the tuberosities that we'll find in our body. We'll also see that the distal end of the humerus will have a lateral and a medial epicondyle as well. This picture shows us the lateral view of one of our coxal bones. Specifically on this bone, we'll find the anterior superior iliac spine and the anterior inferior iliac spine that can both be found here. These are pointed, slender processes that come out of bones. So hopefully by now you're seeing that many different bones in our bodies will all have different types of bone markings. You'll see that the coxal bone will have spines, crests, tuberosities, and much, much more. So it's very important that we focus on the functional group of how each of these bone markings are classified. Our second group of bone markings refers to our projections that will be useful in forming joints. We have 4 of these projections, namely heads, facets, condyles, and rami. Heads will often be found on our long bones. These typically tend to be round and oftentimes are attached to a very narrow neck to increase movement at those particular joints. Remember, these structures are now going to be useful for joint formation. So these are the processes that come out of bones that aid in articulation. One of the best examples of a bony head in our body is the head of the femur. You can see this is the head of the femur here, and it's attached to a very narrow neck. This increases mobility at the hip joint. We've already seen a couple examples of condyles, but condyles typically tend to be rounded projections at the locations of our bones where they will articulate. Our examples of condyles were showing us the medial condyle and the lateral condyle of the femur. These bony projections in particular will come together to form different joints with other bones. And oftentimes condyles will have epicondyles that can be found just above them. Facets are very, very important bone markings for us to understand. Facets refer to smooth articular surfaces. And oftentimes we'll see two bones, each with a facet that will contact each other. Specifically with the vertebral column, we'll see lots and lots of facets that can see on these bones, as there are many places where these bones articulate with. We'll specifically find the transverse costal facet over here, and we'll find the superior articular facet shown here. The coxal bone has a very large facet that's known as the acetabulum. This is given its own name, but it is in fact a flat, smooth articular surface, so we would classify this structure as a facet. This in particular is where the head of the femur will articulate with the coxal bone. Our last projection for joint formation is referred to as a ramus. A ramus can be described as an arm-like bar of a bone. In particular, we'll see a ramus down here and also up here on this bone known as the mandible. Our last group of bone markings refer to the depressions and openings for different vessels. Instead of being prominent processes that stick out of bones, this group includes structures such as indentations and holes that we can find in our bones, and we'll see that this is one of the more diverse groups that we'll find. In particular, some of the depressions and openings that we're going to discuss are a meatus, a fossa, grooves, fissures, and foramen. So first and foremost, many of these depressions and openings are very similar. A meatus refers to a canal-like passageway, while a foramen refers to a hole. Foramen can be very large, but they can also be very small. And there are some meati on the body that are larger than some foramen, and some meati that are smaller than other foramen. So in general, a meatus is more so describing a deep hole, while a foramen is referring to more of a shallow hole. Fissures, on the other hand, are also very similar structures, but these holes tend to have very narrow openings. Let's take a look at some examples of each of these three so that we can hopefully distinguish what these three terms are. The first that we're going to look at is a meatus. This structure that we're looking at is known as the ear canal, but its anatomical name is known as the external acoustic meatus. This is often sometimes called the external auditory canal or the external auditory meatus as well. But this is a very deep canal-like passageway, and therefore we classify it as a meatus. One example that we can see on this slide is the obturator foramen. Remember, a foramen is a hole, and foramen are truly one of the most diverse bone markings that we will find on the body. There are very large foramen and there are very small foramen. So generally, you want to understand that a foramen is some type of hole that can be found in a bone. Fissures, on the other hand, are holes that can be found in bones, but they are very unique in their shape. The superior orbital fissure can be found back here. This is a very slit-like and narrow hole, so therefore we term this as a fissure instead of a meatus or a foramen, based on its shape. One of the most prominent foramen that we can find on the body is the foramen magnum. This is the large hole that can be found at the base of the occipital bone that will allow for the projection of the spinal cord into the vertebral canal. We will specifically learn to identify this foramen when we get to the axial skeleton. So meati, fissures, and foramen are all very similar structures, but get their names based on their shapes. Now fossa and grooves are our last two bone markings that we need to be aware of. Fossas are basin-like structures. Essentially, fossas are very wide indentations in bones that form an impression in a bone. One example of a fossa that we'll find is on the ilium, which is known as the iliac fossa. You can see from this picture that this portion of the bone is impressed into itself. And if we were to position this just right, this would be able to hold a liquid. So fossas can more accurately be described as impressions that can be found in bones, and will often have other structures that will come into fossas and attach at these locations. Our last bone depression are grooves. Grooves are elongated trenches that can be found on bones. These don't typically tend to be very, very deep. However, this is one example of a groove that we will see. This is the radial groove that can be found on the humerus. So meati, fossa, grooves, fissures, and foramen are all of our depressions and openings. I'll just say one more time that it's very important that you can classify each of these bone markings into their respective groups. As we will come to look further at the axial skeleton and the appendicular skeleton, we will start to learn some of these bone markings that can be found on specific bones. But for now, I just want you to have a general understanding of what each of these structures are and which group they reside within. One other bone structure that we will find associated with the skeletal system are sinuses. You've likely heard of sinuses before as the cause of congestion. But sinuses do a little bit more for our body than just get stopped up. Sinuses can be defined as spaces that can be found in bones. And sinuses are lined with mucous membranes, as they are considered open cavities and they connect to the external environment. Sinuses serve two purposes for our body. They will lighten bone and also add resonance to the voice. These spaces remove lots of extra weight that would have been placed on the skull. And without sinuses in general, you might always talk like you do when you're sick. So we're now going to switch gears a little bit and start looking at the histology of compact and spongy bone. Compact bone will look quite different from spongy bone under the microscope. So we're going to take some time to learn how to distinguish both of these types of bones from each other. On this slide, we're looking at a section of compact bone. And when I look at compact bone under the microscope, I see lots of chopped down trees. Compact bone in particular has a functional unit known as an osteon. An osteon refers to this cylindrical structure that can be shown here. We'll also have another osteon over here, and over here. Compact bone will be ridden with osteon. And all of the osteon of our compact bone will share the same general structure. In the center of the osteon, we'll have what's known as the osteonic canal. You can also call this the central canal or the haversian canal. The second name for osteon is known as the haversian system. That's actually the way I learned osteon, so you can call them either or. But the osteonic canal or the central canal will also do just fine. In particular, the osteon is going to have these tubes of bone matrix, known as osteoid, that are wrapping around each other in different layers. These bone matrix tubes are known as lamellae, and we can roughly see their appearance as their layers will continue out as we go towards the edge of the osteon. Now these little dark spots that we can see in various places of the osteon are referring to osteocytes. Osteocytes are mature bone cells that monitor and maintain bone homeostasis. Osteocytes will sit in little spaces known as lacunae, and we can see many osteocytes that are spread throughout this osteon. We'll also have these little tiny tubes known as canaliculi that will connect the lacunae of different osteocytes. These canaliculi are essentially these very small channels that give osteocytes a means to communicate with each other. Osteocytes will have long, slender projections, and they will stick them through these canaliculi and connect to one another. So this is, in fact, the functional unit of compact bone. Spongy bone is going to look a little bit different. It's going to look a little bit more disordered, and we're not going to have structural osteon in this type of bone. However, we will have osteocytes that sit in their lacunae, and we can see those shown around here throughout the trabeculae of the spongy bone. That's what's shown in all of these reddish portions. Now, in between the spaces of the trabeculae, we must remember that we'll find red bone marrow. Red bone marrow is going to be extremely important in the production of blood cells, and you'll discuss a bit more about red bone marrow when you get to Anatomy and Physiology 2. The last topic that we're going to discuss within our skeletal system introduction are the skeletal cartilages. Cartilage is also described as a type of supportive or hard connective tissue. It's not quite as hard as bone, but it does offer lots of support to our body. Unlike bone, cartilage is avascular, and it does not contain any nerves. This is one reason why it takes cartilage a long time to repair itself, because there's not a lot of blood supply taking nutrients to our cartilages. Just as bones were wrapped up with the periosteum, cartilage will be wrapped up in its own connective tissue layer known as the perichondrium. Chondro is our anatomical term for cartilage, so we'll see that our chondrocytes refers to our cartilage cells. We have osteocytes that are found in bone and chondrocytes that can be found in cartilage. We will come to see that we have three different types of cartilage in the body, namely hyaline cartilage, elastic cartilage, and fibrocartilage. And while each type of cartilage will look different under the microscope, all of them will have in common chondrocytes that sit in the lacunae of the cartilage. Let's take a look at our first type of cartilage, hyaline cartilage. Of all of the cartilage in our body, hyaline cartilage is the most common and the most abundant type that we will find. Under the microscope, hyaline cartilage appears rather glassy, and that is because of the fine collagen fibers that make this type of cartilage up. Collagen is one of the most common types of proteins that we will have in our body, and this protein is going to play a major role in constituting the structure of cartilage. Now, I mentioned on the last slide that we have hyaline cartilage, elastic cartilage, and fibrocartilage. Each type of cartilage offers us something different in terms of flexibility and resilience. Some types of cartilage offer us more flexibility, and other types of cartilage offer us more resilience. Hyaline cartilage is a nice middle ground in terms of flexibility and resilience. It offers us both flexibility and resilience due to the fine collagen fibers that make it up. Hyaline cartilage specifically can be found in the nose, making up the costal cartilages, and also in fact at the ends of our bones. Articular cartilage is a type of hyaline cartilage. This is an example of what hyaline cartilage looks like under the microscope. It has a very smooth appearance, and that's why we say that it's rather glassy. Because hyaline cartilage is made up of fine collagen fibers, we will not see any of these fibers imposed in the actual matrix of the cartilage. But we will see these chondrocytes that will sit in their lacunae. Once again, hyaline cartilage is the most common type of cartilage that we have in our body. And I'll show you a diagram at the end of this presentation depicting all of the places where hyaline cartilage can be found. Now, elastic cartilage is our second type of cartilage. Elastic cartilage is named because it's primarily made up of elastic fibers. These elastic fibers have the ability to stretch to 150% of their initial length. So these elastic fibers make elastic cartilage very, very flexible. I mentioned that all types of cartilage sit on this spectrum of flexibility versus resilience. Elastic cartilage, while not as resilient as hyaline cartilage, will be a bit more flexible. We find elastic cartilage specifically in two places in the body, and those locations are in the ear and the epiglottis. The cartilage in the ear is extremely flexible, and it needs to be, as the ears are protruding from the body. If the ears were made up of bones, based on their location, it would be quite easy to break them. So the ear needs to be made up of elastic cartilage to provide them flexibility. Now, the epiglottis is a structure that you may not have heard about in great detail. The epiglottis is a little cartilaginous flap that can be found in the throat. If we take a scope and look into the back of the throat, we'll see that there will be two openings. One opening will be to the trachea, which will eventually lead to the lungs, which is where air goes in when you breathe. The other opening is the esophagus, which will lead into the digestive system, which is where food is supposed to go when we swallow it. Both of these tubes, the esophagus and the trachea, are found right next to each other. And it's extremely important that we get food into the esophagus and air into the trachea. If for some reason food enters into the trachea, that can potentially block the airway and can be life-threatening. So the epiglottis is a nice little mediator between these two systems. The epiglottis will cover up the trachea so that no food can enter the respiratory system when we swallow. This is the second structure in our body that's made up of elastic cartilage. Now we can see elastic cartilage on this slide will look quite a bit different from our hyaline cartilage. You can see that all of these fibers in here look rather disorganized. And that's because these fibers need to be in lots of different directions to facilitate flexibility in many different directions as well. But again, the similarity that we'll see with every type of cartilage is that we'll have chondrocytes that sit in their lacunae. The elastic fibers are a bit larger than the fine collagen fibers are, so we're actually going to be able to see those fibers on this slide. We weren't able to see the collagen fibers with hyaline cartilage because those fibers were so fine. But our third type of cartilage, fibrocartilage, we will be able to see the collagen fibers. Fibrocartilage is on the other end of the spectrum in terms of our cartilages. This is a very resilient type of cartilage that does not offer us as much flexibility. Fibrocartilage is very compressible, and that's why we'll find this cartilage in the intervertebral discs, in the menisci of the knee, and also in one other structure known as the pubic symphysis. The pubic symphysis will connect the two coxal bones down at the pubis bones. In order to make fibrocartilage a bit more resilient, the collagen fibers that make this type of cartilage up will be much thicker. We can see on this slide fibrocartilage and the very thick collagen fibers that can be found within it. You can see these fibers here are much thicker and therefore we'll be able to see them under the microscope. You'll have your chondrocytes that sit in their lacunae and be able to distinguish this type of cartilage based on the more regular arrangement of the fibers compared to the elastic cartilage. So taking what we now know about each of these types of cartilages, we can see on this slide the prime locations where each of these cartilages can be found. Hyaline cartilage was in fact our most common and most abundant type of cartilage that we have. Hyaline cartilage can be found at the ends of our long bones, making up the articular cartilage. It can be found in the nose, and also here composing the costal cartilages. Elastic cartilage can be found in the ear and also in the epiglottis, which we will not see depicted on this slide as the epiglottis has been removed. Lastly, we'll have fibrocartilages that can be found in the intervertebral discs, in the pubic symphysis, shown right here, and in the menisci in the knee. Again, we must keep in mind that these cartilages exist within a spectrum. Fibrocartilage is going to be the most resilient type of cartilage, but offer us the least flexibility. Hyaline cartilage, on the other hand, is a nice medium between fibrocartilage and elastic cartilage in that it provides us both resiliency and flexibility. And then elastic cartilage is on the other end of that spectrum, providing us less resilience but more flexibility. So make sure that you have a good idea of how to identify each of these types of cartilages under the microscope and know the locations where each of these cartilages can be found. That will conclude our introduction on the skeletal system. I will see you all in our next lab recording when we will start to cover the axial skeleton.